today I'm going to talk about viruses. I chose this topic because of the virus emergency. My background in viruses is my bachelor's degree was in microbiology, which includes virology. And in my professional life, I played with several different viruses. In fact, I might talk about them tonight, some of them tonight. First of all, do you guys think a virus is alive? Who thinks a virus is alive?
Just a virus?
A virus, yeah. Is it alive? Is it dead? Who thinks the virus is dead?
Sometimes.
Who thinks it's like Schroeder's cat, dead or alive, dead and or alive? That's how we consider a virus, because it does not have any aspects of life. It doesn't metabolize, it doesn't reproduce itself, it doesn't have movement, has no cellular composition, no organelles.
It doesn't reproduce?
Not by itself.
Not itself.
Not by itself, it doesn't.
Well, does anything by itself?
And this is my favorite virus.
Oh.
What is that?
This is a T4 virus, spider virus. And this is my favorite virus because it looks pretty cool. This is a high quality electron micrograph of that
That one
virus.
doesn't have as many.
Is it a virus that infects spiders?
No?
No.
No.
What does it do?
That one only has 6 legs, the other one has 8.
This is a more primitive electron micrograph because people don't believe that's real, right? Do you believe that's a real electron micrograph? That is a scanning electron micrograph.
Yeah, it does.
This is an electron micrograph from way back in the old days of that virus.
What does it do?
This virus does not infect humans, does not infect animals, plants, fish, reptiles. It affects only Bacillus bacteria.
Oh, yeah. Oh, I've heard of this.
And this is the virus that you use when you're learning about viruses. This is a virus that you use in Virology 202 laboratory. But it does have a lot of features of animal viruses. And it's safe to use, which is why we use it. And one thing that it has in common is it has the capsid here that encodes the nucleic acid. Okay, and this one has double-stranded DNA in its capsid. Now this body here is not common to most viruses. That's why I like this virus. It looks like a spaceship.
I am nomad. I am perfect.
And if this virus got into a culture of bacillus, you'll see what's going to happen.
So small compared to it.
Oh.
This is one of the viruses that can actually move.
This is incredible.
Ooh.
It hurt. Ooh. Yeah. What a reaction.
Oh, look at this.
Oh,
And
man.
mass
And
reproducing.
the virus takes over the cell's apparatus to reproduce itself.
Ah.
Oh, wow.
How quick is that?
Whoa.
In bacteria, it's pretty quick. That would be just an hour or two to get enough viruses to explode that bacterium.
Wow. Was that actual microscope?
No,
video?
this is animated.
Okay.
And all those little guys get out and they float around until they find more bugs to infect. And we've actually discussed using these types of viruses because there's a bunch of viruses that infect only bacteria, using them for highly resistant bacteria infections in humans. The problem is that it's a big chunk of protein, so your body makes antibodies to it real fast, so you couldn't use it very long. So anyways, the life cycle we just saw was that the virus injected his DNA into the cell, and the DNA got busy using the cell's apparatus to make proteins and more DNA, and then it packaged them up, and then the cell exploded. That's called a lytic cycle, because the cells lyse. And in animal cells, some of the viruses lyse the animal cells as well, some of the animal viruses.
It's L-Y.
Lyse. And the other thing that they can do, which is also common in animals, humans and other animals. Okay, here's the situation. This cell gets infected. It goes down. It has two choices now. Sometimes the virus will start making more copies, and it'll be a lytic cycle. Okay, it lyses. Sometimes it'll go through what's called a lysogenic cycle, where it will actually integrate itself into the genome of the bacterial chromosome. And then what happens is the bacterium can grow basically indefinitely or until something triggers that virus to become activated. Okay, and when that virus becomes activated, it goes back down through the lytic cycle. Okay, does everybody understand that? And in Virology 202, what we would do is we would be given a sample of virus and have to determine the titer. So you dilute it out several times, and you would plate it out onto a lawn of bacteria and let it go for a day and count the plaques that form. So that was a cool virus, and that was the first virus I ever played with. And I still think it's the coolest virus. This is it compared to some human viruses. It definitely has a nice design to it. Okay, there is another way that viruses infect cells. And this is for animal cells, not bacterial cells. And I have a little video here that gives you a nice idea about it.
Every cell in your body is a tiny factory, constantly making proteins. Your genetic code, or DNA, directs every activity. Protein-making machinery in the cell reads the cell's own DNA and makes sure the cell proteins go where they're needed. Viruses have their own genetic code, but they don't have the machinery to copy their DNA. So viruses need your cell's machinery to reproduce themselves. A virus starts this process by using the keys on its surface to gain entry. Like a Trojan horse, it enters the cell. The virus releases its own DNA into the unsuspecting cell and hijacks the protein-making machinery. Now, this vital cell function becomes an assembly line for new virus particles. No longer in service to your body, the cell now serves the virus, manufacturing thousands of new virus particles, which are released to enslave more cells, thousands and thousands more.
And what this virus was doing, it was budding out of the cell. Okay? And viruses that bud out of the cell take some of the cell membrane with them. Okay, and that's one of the ways that they try to evade our immune systems, is they coat themselves with part of the cell membrane. They insert some viral proteins into it because they need some viral proteins to be able to attach to the cells and to get inside of the cells and out of the cells. This should be taught to creationists because viruses have evolved all kinds of different ways of getting their genetic code into cells, and they use all different types of genetic codes to get there. There's double-stranded DNA, there's single-stranded DNA, there's double-stranded RNA, and single-stranded RNA. And in 1972, David Baltimore, who was a Nobel Prize-winning scientist, biologist, came up with this categorizing of viruses based on the DNA or RNA that's inside the capsid. Okay, so we got the first group is double-stranded DNA, and in that group are some viruses you probably heard of. Herpes virus. You've heard of herpes. Everybody here has heard of herpes virus, and I can guarantee you all have had it, and probably most of you still have it. because herpes virus is a DNA virus, and it's one of those viruses that integrate into your cells.
Chicken pox.
Chicken pox is an example. Yeah, herpes zoster
Shingles.
gives you shingles when you get stressed out. Yeah, when you immunocompromised, you get shingles. There's another cytomegalovirus, which is something that really doesn't make us sick, but everybody gets exposed to it, and some of us carry it around with us. And it can come back to haunt us when we get old and are immunosuppressed, for sure. Epstein-Barr virus, the kissing disease virus, mononucleosis. Everybody's exposed to that.
Isn't that always, also chronic fatigue syndrome virus? Is that the same virus?
It's just temporary. It makes you tired, yeah. Yeah, it makes you tired. But you usually get over that. But you don't usually get over the virus because it integrates. And the problem with the Epstein-Barr virus is it can cause cancer. It's a cancer virus that most of us carry with us. The other virus in this group that you probably heard is papillomavirus. And again, everybody has been exposed to papilloma. When you're in school, your friends that have warts, you're exposed to that, right? When you get older and you have sex, you're exposed to the papillomavirus through sex. And the papillomavirus, the sexual part of the papillomavirus can be passed to children during childbirth. And the problem with papilloma, of course, everybody knows it causes cervical cancer, right? Group 4 and 5 are single-stranded RNAs, okay? And the one that says positive, see the one that says positive up there?
Yeah.
That's where the coronavirus is. It's a positive DNA, which means RNA, which means it acts as messenger. It's messenger RNA. That gives coronavirus a big advantage for infecting cells because it injects messenger RNA right into the cytoplasm of a cell. And the negative RNA up there, you see a negative RNA?
Mm-hmm.
That is the flu virus. And both the flu virus and the coronavirus bud through the membranes to disguise themselves. That's one thing that makes them very dangerous. And they both have very efficient ways of getting their, making their proteins and getting their genes reproduced in the cells. 6 is retrovirus, single-stranded RNA, but it carries an enzyme called reverse transcriptase. This is where the AIDS virus is, right, in that group right there. There's also a virus in that group that's called HTLV. Has anybody ever heard of HTLV? Human T-cell leukotropic virus. Well, you probably heard that term because when the AIDS virus was first discovered or uncovered, they put it into the HTLV categories. There's several HTLV viruses, and they put it into that category for a little while, and they took it out. So for a while, the news media was talking about HTLV. But there's other viruses in that category. One of them is HTLV1, which is a virus I worked with when I was at the Cancer Institute. And this virus this virus can integrate into your cells. It's probably in the blood supply because it's not tested for, okay? About 20% of the population carries it, and it causes non-Hodgkin's and Hodgkin's lymphoma, and you can transmit it sexually as well. But you never hear about this virus because this virus doesn't do anything to you until you get old and you become immunocompromised. In fact, when I'm watching the news at night and I hear some old movie star or somebody died of leukemia at 92 years old, it's that virus. I know it is. Okay, so anyways, I wanted to impress upon you that viruses and cancer are heavily linked together. And we're finding more and more and more about how viruses cause cancer. So anyways, there's lots of oncogenic viruses, and we're finding more all the time. But today I know you want to talk about respiratory viruses, right? Okay, so this is a list of viruses and one bacteria, by the way, that causes respiratory problems in this throughout the year, and some of them are seasonal. Now, rhinovirus is not enveloped; it's an RNA virus, and that's the most common cause of colds. Okay. Coronavirus is an envelope virus, and it tends to be a seasonal virus because envelope viruses are easily disrupted. That envelope, that lipid envelope that they take from the mother cell when they bud out, is easily disrupted. So coronaviruses do not like sunshine, they don't like warm temperatures, so they don't tend to be a problem in the summertime. PIV is parainfluenza. Those are actually enveloped viruses, but they're hardier for some reason or another. RSV is not encapsulated. RSV is respiratory syncytial virus. Influenza is seasonal because it also is encapsulated. So anyways, evolution, if you want to get a quick lesson in evolution, you just have to look at viruses for sure. This is the flu virus. We'll start out with the flu virus, okay? So the flu virus has its RNA in here. It has a protein coat, and that protein coat is encapsulated in the membrane from the cell that it budded from. And it's got two antigens, two proteins that it puts inside, that lets stick out of that membrane. Hemagglutinin is one of them, and neuraminidase is the other. And you've heard the terminology for this. I know you have, H1N1. So that's where this comes from, okay? Because those are what we mutate, and those are what changes with this virus. And I started out with this virus because of the fact that it can kill a lot of people. It kills on a typical year 20,000 to 100,000 Americans, okay? So far, how many people have died from this coronavirus? 4,000? That seems like nothing compared to what the flu does. And we don't get crazy about the flu because of the fact that we have an immunization for the flu. So that eases everybody's minds about the flu, I guess. Because we don't talk about it too much, except for get your flu shots. So anyway, so far this year in the United States, there's been over 16,000 deaths caused by the flu, and it could be as high as 30,000. And the reason we have such a hard time figuring this out is that they're not reported properly. As you can imagine, one of the most common ways to die from a respiratory virus is to get a secondary bacterial infection. So you go to the hospital and you die, and they say bacterial pneumonia, right?
Yeah.
We grossly underestimate the amount of viral deaths that there are.
What do you think is the best month to get your flu shot since
October.
But 16,000 people already died.
Since October. Because usually the virus starts to raise its ugly head towards the end of October. And we've had some horrible flus, okay? The flu of 1918, everybody has heard about that. And if you haven't, I have a quick video about that. The H1N1. The H1N1 came back again too in 2009 and caused some damage. It was noteworthy. Definitely helps to have immunizations. This is an old graph of mortality during the 1918 pandemic. And you can see 3 bumps, really, if you look. And the CDC made up this graph of the 2018 pandemic. And you can see 3 bumps. Late spring, early summer, winter. And this is a graph from 2006 to 2009. And you can see that the bumps, how they go away and come back and go away and come back. And that's what I'm a little bit worried about the coronavirus, because we really haven't had a pandemic with the coronavirus before. And it could be that it's going to be scare us right now, but when the summertime comes, it kind of will die back and then come back with a vengeance in the wintertime. But anyways, in 1918, it was pretty sad. So many people died in the war, and more people died from the virus. And I have a quick video.
An estimated 500 million people infected, between 50 and 100 million dead. A world torn apart by war and by a strange, deadly new disease. It swept across the world, leaving nowhere untouched. It was so deadly, rumors spread through the streets that the Black Death had returned. This was the Spanish flu of 1918, the deadliest pandemic of the 20th century. Epidemics have infected societies throughout history. Usually epidemics come and go, but some truly stand out for their viciousness. In recent history, there has been no pandemic more devastating than the flu of 1918, sometimes called the Spanish flu or swine flu. It killed about 675,000 in the United States alone. The outbreak came in the midst of World War I, and the flu would actually kill more troops than combat. The flu of 1918 was so deadly it killed more Americans than World War I, World War II, Vietnam, Korea, and the wars in Iraq and Afghanistan combined. Yet Spanish flu was horrifying for even more reasons. It was very contagious, and it killed extremely quickly, sometimes in as few as 12 hours. It killed people in the prime of their lives, with the most victims being young, healthy people aged 20 to 40. Additionally, it killed some of its victims in bizarre, brutal ways, completely unlike a typical flu virus. Some symptoms actually closely resembled the plagues, which killed millions in the Middle Ages. But scientists have analyzed the pathogen's genome and verified it was not Black Death which killed millions in 1918. It was just the flu.
The coronavirus is named SARS-CoV-2. And that was going to be the name of the disease as well, but the SARS part scared people so much that they changed the name to COVID-19. But you'll still see the virus as described as SARS-CoV-2. And the SARS virus is very similar to the flu virus. It has RNA in the center, single-stranded RNA in the center, has a protein coat, it has the cell membrane around it, it has hemagglutinin protein, and it has S protein. And that's what makes it different, the S protein. The hemagglutinin is similar, not identical to the flu virus. This probably came from a bat. The sequence says that it probably came from the bats. It may have come from the bat into other animals to us, but it was a bat virus to begin with. And the reason it's kind of nasty for us is because the bats have a different type of immune system that they had to adapt to. They had to divide faster in the bats to keep up with the bat's immune system, which is why it's becoming a problem. One of the reasons it's becoming a problem for us. This is symptoms, and you can see that one of the symptoms that is different from the cold and the flu is shortness of breath. And even the people who don't have severe disease describe feeling like they're short of breath, or they can't get enough oxygen. And this is a better description of some of the symptoms. Everybody here has been infected by a coronavirus at some point, because coronaviruses cause about 20% of common colds. Okay, but that's not the same kind of coronavirus that we're fighting right now. But if you read in the papers or seen on TV, kids are not having a problem with this virus, because kids have so many viruses, being in school and preschool and stuff, that their immune system is hyped up, and they see coronaviruses, and there's enough similarity between some of the coronaviruses, they don't get very sick from this virus. Oh, this is how you die from the coronavirus.
Sure. So I hear this question a lot. How does the coronavirus actually kill people? There's a recent article in The Lancet that showed that of 41 people that were admitted to the hospital, 6 of them died, and all of them were on ventilators, and they died with something called ARDS. And ARDS is how the coronavirus kills. It's not just the coronavirus, but many other viruses, including the influenza virus that we have every year. How is it that this happens? It's through acute respiratory distress syndrome, and I'm going to explain to you how that happens. So you first have to understand lung anatomy, and to understand that, I like to show you a tree. So a tree has a tree stump, and then it branches, and then those branches branch, and then further those branches branch, until finally you get to the leaves. And these leaves capture the sun's rays, and that's what gives you photosynthesis, and that's how the tree lives. And so what happens is that this tree and the branches increase the surface area of the leaves on the tree, so that if you were to pluck off all of the leaves and you were to put them on the ground next to each other, the surface area that is represented by those leaves would be larger than the shadow that is produced by the sun on that tree. Well, it's the same exact thing that happens with your lungs. You've got an airway, and then that airway divides into a right mainstem bronchus and into a left mainstem bronchus, and then you have a right upper lobe, you have a right middle lobe, right lower lobe, left upper lobe, and you have a left lower lobe. So this is the left side, this is the right, because you're looking at the patient. And then these things, of course, divide into much smaller branches. And instead of leaves at the end of all of these things, what you have is something called an alveoli, which is a tiny little small grape-like structure that the air gets into. And the air, of course, has oxygen. So what does this look like on a large scale? Here's what an alveolus looks like. How many alveoli are there in the human body? Well, there's about 600 million of them. These are very, very small. So what happens is deoxygenated blood comes by, and its job is to pick up the oxygen that comes in to the alveoli. And then when that oxygen comes in, it oxygenates the blood, and then that blood goes back to the heart and then to the body and all your muscles. That's how you get oxygen. And so you can imagine that this is very, very thin, because the oxygen which comes down here has to diffuse into the bloodstream. So far, so good. But what happens? Well, just like when you hit your finger in the door, your finger swells. That's because there's inflammation occurring where you hit your finger in the door. And inflammation causes a leakage of fluids into the tissue space. So what happens here is that you get a viral infection. The virus affects your lungs. And with ARDS, the entire lung becomes inflamed. Not just in one area like you would have with a pneumonia, or one particular area, for instance, on your finger, and it would just stay in one particular finger and your whole hand wouldn't swell. No, with ARDS, the entire lung goes crazy with inflammation. And so what happens here, instead of having a nice thin area, inflammation goes everywhere, and you get a large barrier of fluid that goes into the interstitial space. Furthermore, these capillaries start to become leaky, and fluid starts to leak into the alveolar space as well, and this starts to fill up with liquid, proteinaceous liquid, liquid that prevents oxygen from getting into the bloodstream. And so instead of having nice oxygenated blood, this blood becomes hypoxic, and you become hypoxic if you have ARDS, and you have a hard time breathing, and that's when you get placed on the ventilator. There's really nothing you can do to speed this up. There's nothing that you can do to slow it down. You have to be supported on the ventilator so that you're getting enough oxygen and that the machine can breathe for you until, just like everything else, after you hit your finger in the door and the swelling goes away, this fluid will eventually go away as well. The key, though, is keeping you supported during that period of time until the fluid goes away. And then, once again, the oxygen will be able to go back into the system, and you will get oxygen back to your tissues.
Okay, now with this virus, one of the problems with it is that it does so much damage that you just can't be on a mask of virus. You have to be intubated when you get serious with this virus, which means they have to put a tube down into your lungs. Okay? So that's one of the problems with this virus. And it can happen with flu too, but this virus is a little bit more aggressive. And this is not the flu virus, okay? This is not. People are trying to make this out to be similar to the flu virus. And it's not really. And one major reason that it's not is that Americans have immunity to the flu. Americans almost all have immunity to the flu because 40% of us are smart and get immunized every year, and the ones that don't get the natural flu. So we're always carrying a little bit of immunity to the flu virus. There is no immunity for this in the population for this virus. So if it really takes off, there's going to be a lot of damage done. There's going to be a lot of deaths from this virus, for sure. And this is a small number right here. I mean, this is assuming that it was like the flu. But it's not. So instead of only 30 million people getting it, like who gets the flu every year, 300 million people could get it. And the death rate is definitely higher than the flu. We think is higher than the flu. Right now, the death rate is calculated to be about 3.6% worldwide. Okay. In Fort Myers, it's 50%. In Florida, it's 14%. But we don't know for sure what the mortality rate is because we're not doing enough testing. And if we tested more, and especially if we go and test anybody that has any kind of sniffles or any kind of symptoms, we might see that the mortality rate is not all that high. But then again, we might not. We don't know yet. We just don't know. But in Singapore, they've had like 150 cases, and they have had no deaths so far. So. And they're testing more in Singapore. So they're seeing the cases that are not very severe.
Okay, so how do you kill that virus?
Don't touch your face. Wash your hands. Stay 6 feet away from each other. Stay home. I mean, you don't want to get this virus. People need this. If this virus gets entrenched in Fort Myers, you need to stay home. Okay. Right now, I don't think it's entrenched in Fort Myers. We had 2 cases. They were people who returned from vacation and got sick. They exposed, who knows, probably not a lot of people. And I know they traced the people that were exposed and have them isolated. So not to say there couldn't be people walking around with not very serious case of it, spreading it around.
Okay, but John, you mentioned earlier that the virus doesn't like sunlight. So when we now go on the beach, if I'm laying in the
You're
sun,
fine going on the beach. You're fine going for a walk in the woods. Just don't.
But it
Just
would
don't.
help to prevent or give a better immune system to fight the virus? Or
No, there is some evidence that getting into the sun a little bit does help your immune system. Yeah. But being outside in the sun, talking to people, you're probably not going to get the virus. Being inside and somebody sneezing, that's how you get the virus, or coughing. If there was a vaccine, the average American wouldn't get it. Just like the average American doesn't get the flu vaccine.
40%, you said.
40% get the flu vaccine, yes. But there's anti-vaxxers who think that the natural infection is better for you, which is not true. We saw all the ways that viruses can integrate into our genome, and we can carry them around, and they can do damage to us later. Get vaccinated. Okay, so there is no vaccine yet. There used to be a vaccine for a dog, but that was a different kind of a coronavirus. They did try to develop some vaccines for the SARS and stuff, but that was also a different coronavirus. Let me explain to you vaccine production very generally. The old tried and true method is that we grow up a whole bunch of virus and kill it and inject it, and that's your immunization. Now the problem with that method is you have to grow up huge amounts of very dangerous viruses. Then you got to kill them, and you got to validate that you killed them enough so that they're not going to infect somebody, and then you have to make sure that the antigens, or the immunological part of it that stimulates the immune system, is still intact. The other way to do it is to go into nature and look for a similar virus that's not as deadly, that you can use. A good example of that is the virus for smallpox was from a cowpox virus, right? And it works against smallpox. The other thing that you can do is you go in the laboratory and you can make a virus that's less infective or less deadly. And that's called attenuation. You can attenuate a virus. And there's the polio virus is an example of that. And the third way, and the new way, and what could become the standard way, is to make messenger RNA from the virus that you want to immunize against, and inject it into the blood system of the people you want to immunize. And some of that messenger RNA, which will be coated with something to protect it, will get into some of the cells. And you want to target the immune cells, because in the immune system there's certain cells that are called antigen-presenting cells. They'll take foreign antigens and process them and put them out on their surface and present them to other cells of the immune system. But even just regular cells, if they can express that protein, will help stimulate the immune response. And that's in experimental trials right now, and there are no, as far as I know, there are no messenger RNA vaccines yet, but this would be a good way to use it. You could make them really fast, because you wouldn't have to grow the virus. You just have to synthesize the messenger RNA, which is pretty easy nowadays. Oh, I have a immunization movie for you tonight, because it shows how our modern society is based upon immunization. And that's one of the reasons this virus is driving us crazy, because we don't have an immunization for it. So most of you were around when some of these immunizations were developed, I know. I can remember my mom dragging me downtown, my little brother and I downtown to get the Sabin polio vaccine, the attenuated live vaccine. And the line to get it at the city hall, it was given out for free, went way back into the neighborhoods. I didn't even know there was that many people in my hometown. But that was how worried parents were about that virus. And now we take it all for granted, except for the anti-vaxxers. Okay, so antivirals. You don't hear too much about antivirals because there isn't much on a virus you can target. You know, antibacterials. Bacteria are fully functional cells. They have ribosomes that are different than our ribosomes. We can attack the ribosomes. They have cell membranes, and internal membranes that are different than ours. We can target those. On a virus, there isn't as much to target. But that being said, there's a lot of antiviral drugs that are in development, and there are some that are being tested for the COVID-19. And in fact, this one is actually in phase 3 clinical trials.
It's too small, you're going to have to read them off if you're going to.
Remdesivir. Yeah.
You saw that one coming.
This one here is in phase 3 clinical trials because it has already been tested on viruses and was found to be safe. And now we have to test it to see if it's effective on this virus. And it's called Remdesivir, and it's from Gilead Pharmaceutical. And it targets RNA-dependent RNA polymerase. And there's another one from a different company, Pavivir, which targets the same thing. And here's a little bit of cell biology, okay? This is the cell membrane right here, okay? This is the nuclear membrane right here of a cell. This is the virus. The virus comes in, it leaves the antigens on the surface of the cell because the membrane fuses with the membrane of the cell, and that becomes part of the cell's membrane again. And it puts its RNA into the virus, into the cell. And this is a coronavirus, so it has a big advantage because its RNA is messenger RNA, and immediately the ribosomes bind to that RNA and start making protein, right? Starts making protein. And the first protein that it makes is the RNA-dependent RNA polymerase, which is also known as replicase. So, and that is about the only thing that we can target, other than the antigens on the surface of the cell, and that's what you do when you do immunization. Now this antigen right here is what it binds to, which the virus binds to attach to the cell. And the receptors for these are very prevalent deep down in our lungs, which is one of the reasons this virus is a problem. So, I mean, one of the big problems in the United States is going to be the government. Because the government has to respond seriously to this. And I don't think that Trump has been very serious about this. And the CDC kind of dropped the ball on this. And I think that a lot of people are afraid of pissing off Trump. So that worries me right there. And the test kits were not sent out in time. I mean, we had 3 months notification. The Chinese gave us the sequence for that RNA 3 days after they got it themselves. And they said, make some test kits, get ready for this virus. And we weren't ready. And that's the main problem I see in Fort Myers right now, is that the people who have been diagnosed were very sick. There could be people out there who are not very sick, who are spreading this virus, because we just don't have the test kits. One of my friends is a microbiologist in town for Lee Health, and he always keeps me informed about what viruses are around. He doesn't have a test kit. There's no test kits in the hospitals. They have to send it out. They send it out to the county. I'm not sure the county has test kits, so if they don't, they have to send it to the state. And if the state doesn't, they have to send it to the CDC. So that's been the biggest problem in the United States, is testing enough people who are around the people who are getting sick to make sure that they're not carrying it. They're not asymptomatic. Yes?
Some that are sick are going to the doctors. I know a couple of individuals where they're testing them for flu A or B, at least, you know, making that determination.
Yes, you can eliminate flu A and B right away. That's one thing you can do, is say, okay, well maybe you have the flu. If you don't have the flu, you have to assume it's probably this virus. But there's a 1,000 other viruses though that can get serious. Okay, Florida viruses. This is not any real data. This is okay. So what do we got here? This is where the outbreaks, so-called, have been. And like I said, the death rate here is like 14%. We only have like 14 people who got sick so far. And the ones in Fort Myers, both of them were travelers. And all three of the ones that died were travelers. And Lee Health says that as of today, there hasn't been any new cases for 48 hours.
As of yesterday.
Is that yesterday?
Yeah.
Okay. Well, I got that off the site today, but so they haven't updated it. We also have other viruses we have to worry about in Florida, of course. You know, we haven't had, I don't think we've had a frost in Fort Myers in 10 years, have we?
Frost?
Probably not.
Frost.
Since I lived here, there has been.
Who said that? There has been?
The first or second year, because it was frost. I thought you never got it either.
No. Usually we get a little bit of a frost. We haven't had any frost for a long time. When I first moved here, there was 2 little frosts every year for the first 8 years I was here. Then all of a sudden, there wasn't any frost anymore.
We had snow in May.
So the frost kills the viruses?
So the frost kills the vectors that transmit the viruses. Yeah.
So do the mosquito sprays.
Yeah, mosquito sprays do too.
That's what they're using.
Yeah, and I mean, there used to be in the wintertime not as many mosquitoes. And there's more mosquitoes now, which is dangerous because these viruses are transmitted by mosquitoes. Dengue fever is a hemorrhagic fever virus. It makes you pretty sick. You know about chikungunya? That came to Florida. Zika came to Florida. Encephalitis viruses come to Florida. Okay. As I was trying to impress upon you in the early part of this lecture, is that DNA can get into our cells, right? From the viruses. Viral DNA can get into our cells. And this happens to us every day, all the time, when we get infections, viral infections. And if it happens to our reproductive cells, then that virus is with us forever. You pass it on to your children pass it on to their children, and they pass it on to their children, and so on and so forth, okay? So, like I said, I would talk about one virus that I worked with, and I think some of you might have heard about this virus before, because I talked about it once before. The first big assignment I got at the NCI was to try to figure out whether or not humans had a virus similar to the mouse mammary tumor virus. In mice, just about the only way that they get cancer is by the mouse mammary tumor virus. Okay? It's a virus that they shed, but it's also a virus that's in their genes that they pass on to their offspring. And this kind of illustrates that. It's an endogenous virus. It's in their genes. They can shed it into their milk, right? They can infect others. It also can cause leukemias in mice. Okay, so my job there was to go in and grow up a bunch of mouse mammary tumor virus and to make a panel of monospecific antibodies to the tumor viruses, antigens. And I could tell you how to do that, but it would take another hour to do that. But once we had that panel, we used those antibodies to stain human mammary tumors. And sure enough, some of those antibodies bound to some of the tumors, suggesting that there is a human mammary tumor virus. And that was almost 40 years ago. And now we have the data through better analysis, because we can make genetic probes now and look at the cells and see if the virus is in the cells. And we've done that, and we can see similar virus antigens. And now we've been able to actually detect the virus in cancer patients, breast cancer patients. So this is a real virus that probably very few people have ever heard of, that we carry with us, and like something, there's a certain percentage of the population that has it endogenously in their genes. So that was one of my viral experiences. The last thing I did before I retired was we were producing a product from a tumor cell line. Because tumor cells are immortal cells, they can grow forever in culture, right? So tumor cells are a good thing to grow biological drugs in. And the FDA says, first thing you gotta do is you gotta figure out if it produces any viruses, okay? The tumor cell that you're using to produce a product. So I was able to isolate a virus off of that. It was a virus similar to this. It was a leukemia cell line. It was probably the exact same virus as causes the mouse mammary tumors. And I grew that up, handed it to the biochemists so they could figure out a way to purify it away from our product. But then also the FDA says you have to have an enveloped and an unenveloped RNA and DNA virus. So I had to bring in all those viruses, develop assays to assay for those viruses, grow those viruses up, purify them, and give them to the biochemists so they could prove that they could purify those away. And that was my last big viral experiment. And that's the end of my talk for tonight.